The symposium now is more about funding opportunities. I was actually talking to Brooks yesterday, and he kind of reminded me that one of the reasons NIH and others attend these meetings, it's a bidirectional exchange, because they're going to tell us about funding opportunity, but they really are here also to listen and to get a sense of what the next steps are so that they can actually put their new RFAs and so forth. So if you haven't had an opportunity to have a one-on-one with any of these individuals, I really encourage you to try and do that. So we're going to start with Dr. Brooks Gross. He's a program director in the Division of Translational Research at the NINDS. Dr. Gross manages a portfolio including the Brain Initiative Translational Device Project in the area of epilepsy, cognition, and neurorehabilitation. He is also the co-lead of the SPARC Hornet Initiative, which is kind of a sister to the program we actually got funded. Please.
Thank you. Can you hear me? I don't know. Yeah? Okay, great. Yeah, so I'm going to talk about several different funding opportunities through NINDS as well as across NIH. It's going to go pretty quickly, but feel free to follow up with me while I'm here or via email, and that'll be on the last slide. So this is just to demonstrate that NINDS's translational and clinical divisions participate in several, or actually run several different programs for translation. We're not going to go into detail on all of them. Some are for neurotherapeutics, some for biologics, small molecules. I'm going to focus on the devices today. This slide is to show that we are trying our best to cover the entire device development and translation pipeline. We have several funding opportunities within NINDS, as well as the Brain Initiative and HEAL Initiative. And there are some things in SPARC, but SPARC will be winding down. So within NINDS, for the early stage research and development, we have partnered with NIMH to explore neuroscience research and early stage development with an R21. And so that's, if you don't know, a high risk, high reward sort of funding opportunity, which requires minimal to no preliminary data. We also participate in the NIH Bioengineering Research Grant, which is an R01, and we participate in both the clinical trials not allowed and the clinical trials optional. And that's for projects that are a little further along. Within NINDS, on my team, we run this Translational Neural Devices UG3 UH3 program, and it's a two-phased award. It's a cooperative agreement where milestones are set. In the first phase, it's for late preclinical stage device development, regulatory approval, and also studies for regulatory approval. In the UH3 phase, you go on to a first-in-human or early feasibility study, so it's a small clinical trial. It can be invasive or non-invasive as long as it fits within the mission of NINDS. Something that I'm really excited about is the Blueprint MedTech program, where we've set up a system or network of resources, the contracted resources that NIH will directly pay for, as well as 2 hubs for the earlier stage development. The hubs are CENTA and Neurotech Harbor, so they're based at Hopkins, and mostly Hopkins and Harvard MGH. And these programs are accepting pre-applications or pre-proposals that are 2 to 3 pages long every 6 months. And that's for earlier stage development as well as some business assistance. So the entry is barrier to entry is pretty low on those. Then it gets down selected to full proposals and deep dives after that. And then we end up funding them based on the institute that it fits within. Blueprint Neuroscience consists of 10 different institutes at NIH, and so there's a variety of missions that a project can fit within. We also have the translational portion, somewhat equivalent to the translational neural devices, UG3UH3. We have a medtech translator, UG3UH3, as well as an equivalent version for small businesses, the U44, which is a fast-track two-phase mechanism. These applications, the nice thing about them is that because we have contracted resources through NIH, you actually don't have to go out and find contractors and get quotes. You just include in your resource checklist the resources you want to take advantage of and what you would like them to do for you. NIH then handles, through our contractor, fulfilling those contracts, and so your budget ends up being a lot simpler when you submit your budget and your application. Some of these, well, actually all these resources are also available through the hubs as well for the earlier stage development projects. So for the BRAIN Initiative, which has been going on longer than MedTech, we have several opportunities that cover earlier stage development through the new concepts, R21, so it's high risk, high reward again. We also have mechanisms to, as you go along that pathway of development for more established projects in the R01 space. We have optimization projects, which is the U01. We've separated out the molecular technologies, so optogenetics, cytogenetics, those sorts of things, into a UM1 now. So we're trying to deviate or distinguish between the devices and the molecular tools. And we do have a robust avenues for dissemination, including this U24, that's a research grant for dissemination of resources. The one that I highlighted in red here is the biology and biophysics of neurostimulation and recording. That's an interesting opportunity to be able to characterize how the different neuromodulation modalities are affecting the system at various levels, and the responses of the biological system to the neuromodulation technologies. Within our team, we have the brain UG3-UH3 and also the direct to UH3. So if you already have IDE approval for an invasive technology to go straight into your first in human study, you can go directly into the UH3 program, which is nice because it allows a full 5 years for a clinical trial, whereas the UG3-UH3 version, you can split between 1 and 4 or 2 and 3 years either way for the 2 phases, but it's a total of 5 years. We do have a brain is signed on with a notice of special interest to the Blueprint MedTech program. So if you have a brain-related project that you want to go through, either the UG3, UH3, or U44, Brain will fund that project through the MedTech program. And finally, for the BRAIN Initiative, I just want to mention the Brain Behavior Quantification and Synchronization Program. This is a really interesting program to use many different technologies, wearables, biologic sensors to understand complex behavior and synchronize that with neural activity to eventually improve how therapy is delivered. And so we do have one opportunity so far with that initiative. Also very quickly mention the HEAL initiative. We have equivalent programs for development for devices. We used to have a U18 when the HEAL initiative first started. We now have an R18, so it's not milestone. And it now includes support for development of devices for opioid use disorder. And we have HEAL signed on also to the MedTech UG3-UH3 translator program and the U44. And we have the Interdisciplinary Team Sciences R01 to uncover mechanisms of pain relief by FDA-cleared medical devices. So that's a little different than the BRAIN Initiative version. BRAIN Initiative version, they didn't have to be FDA-cleared devices. They can be any sort of neuromodulation technique. Whereas with the HEAL Initiative, it does have to be an FDA-cleared device. So the most important slide, I want to thank my team members. We work very well together as a team, and if I can't answer a question, I always can refer you to one of the other team members The email on the bottom there is for our team email inbox, and we check that every day, and one of us, whoever is more appropriate to answer the question, will reach out to you and set up a call and answer any questions and direct you where you need to be. So that's pretty much it for me.
We have time for one question.
I actually have a question. No, I don't.
Go ahead.
Brooks, I mean, considering some of the topics here, autonomic and simulation of peripheral nerves and even spinal cord, can you say a bit about how loosely some of those topics could fit into the programs you list there? Because a lot of them are brain-centric.
Yeah, so the brain initiative is brain and spinal cord centric. So we do extend it to the spinal cord. And I have several projects in my portfolio that don't even look at brain signals. But they're brain-esque because they're looking at circuits in the spinal cord and trying to do closed-loop stimulation to restore function. We also have the HEAL initiative is not limited to brain.
Yeah.
And our NINDS funding opportunities are only limited to whatever fits in the mission of NINDS. And same with MedTech.
Yeah.
MedTech, though, does have a stipulation that it has to be something novel. It can't be something that's already been FDA cleared.
Yeah, and maybe just something specific. I don't want to take the time, but if immune is affecting some brain disorders, right? Too much inflammation in the brain, and there's a technique to modulate that peripheral spleen, whoever's working on that perhaps to say, and then brain imaging of those effects, would that fall under some of these?
I think so.
Yeah,
I mean, I think it helps if you have sort of a rough idea sketched out of what you're thinking of doing, and that helps us better direct you to which of these programs. But I think we've done a pretty good job in covering almost everything you could sort of pitch. So, yeah.
All right.
Thank you.
Great, thank you. All right, our next speaker is going to be Dr. Tyler Best. I meet with Tyler every month. It's been, and maybe back in the summer, it was even every other day. Dr. Tyler Best joined the Office of Strategic Coordination in 2019 to serve as a program officer for the Human Biomolecular Atlas, the HubMap, and the Stimulating Peripheral Activity to Relieve Conditions SPARC programs. And prior to being in the NIH, he worked extensively with DARPA. Welcome.
Thanks, Ziad. While she gets that set up.
Are you going to tell us a joke?
Sure. What do you call someone who tells bad jokes and bad puns?
What?
Dad. I'm glad somebody laughed. While she gets that set up, I'm representing the NIH SPARC, Stimulating Peripheral Activity to Relieve Conditions program. As Ziad mentioned, I joined NIH in 2019 to help manage that. At that point, SPARC was partway through phase one. SPARC is a common fund program. It's not run out of any of the ICs or other institutes or centers, and that means it's supposed to be cross-cutting, multidisciplinary, kind of more cutting-edge research than a traditional IC would fund. And the SPARC program itself, in that phase one, in those early phase that ran through 2021, was focused on mapping peripheral nerves and their innervation of various targets in the viscera, as well as developing technologies and tools to interrogate that system, to modulate that system, and a lot of the work that you've heard over the last couple days was very much part of SPARC or very well aligned with it. There was emphasis on translation, clinical studies, as well as an ongoing resource for data storage, data management that is continuing beyond this SPARC phase 1 past 2021 and into the full duration of SPARC through 2024. Starting last year, we transitioned to stage 2 of SPARC, and we're focused. We realigned, refocused things on some certain aspects of VOX, kind of a new voice for SPARC there, where the V stands for the vagus, where we're hoping to map both anatomically and physiologically the vagus and its impact throughout the body. O, standing for open source neuromodulation, of which the Hornet initiative, which Brooks, as well as Eric Hudak on their team co-lead that effort. And then the Neuromod Prize challenge competition, hence the X, like the X Prize type of competition. So as all government things, we have to come up with acronyms for everything, so we do sub-acronyms beyond sub-acronyms and so on and so forth. So that's where that comes from. SPARC V, mapping the vagus, the human vagus, of which John Osborne's team, Ziad, mentioned. We're funding them to look at the endpoints of vagal nerve stimulation. You know, I was mentioned in the last few days, heavily influenced by indications for epilepsy and depression, but the vagus innervates the stomach, got all the viscera here, and we want to understand what's happening there in addition to the therapeutic impacts that it does for epilepsy and depression. Sistered to that, we are funding two teams at Case Western and at the Feinstein Institute to map the connectivity of the vagus, and that's not just gross anatomy, that's looking down at the fascicular, subfascicular, afferent, efferent levels of the branches throughout the entire body. So it's a very much a painstaking process of doing sections after sections after sections, reconstructing with algorithms for region of interest identification, segmentation, and so on and so forth to map those axons and those fascicles as those fascicles combine, divide, and et cetera, throughout all the branching patterns of the vagus. Next, as I mentioned, the O initiative, or SPARC Hornet, stands for Human Open Research Neural Engineering Technologies Initiative. It's a mouthful. Again, we're funding two teams there. One, again, out of Case Western. It's a different team at our Case, Kevin Kilgore, as well as Ellis Meng at USC, to develop platform technologies that are open source, that are kind of more plug and play, that other systems are able to adapt with and interact with, so that these platforms can be used and hopefully lower the threshold for entry into market and beyond. Lastly, the SPARC X initiative is a prize competition. We are now in stage 2 of that prize competition. We've got 8 teams that are developing solutions to demonstrate independent modulation of more than one or multiple clinically relevant autonomic functions, with the goal of not having or controlling any off-target effects. The data aspect from phase 1 is continuing throughout phase 2 as a support system for the VESPA, the REVEAL team, these vagal mapping, as well as the Hornet initiatives if necessary, as well as it's also open to, as of I think a couple months ago, anybody who has data that wants to deposit it there as part of kind of this initiative to have data sharing plans that the NIH is mandating, of which I think the proprioceptive aspect is looking into with when. So, and that's located here at spark.science. if you didn't know, but that .science isn't actually a URL. You don't have to have .com or .edu or .gov after it. The .science is the domain name. And anyway, learn something new every day. So I am actually, part of my time, I'm an NIH employee, but I'm splitting half of my time helping set up this new organization, this new agency, ARPA-H, Advanced Research Projects Agency for Health, whose goal is to accelerate better outcomes for everyone. It's a agency modeled after DARPA, the Defense Advanced Research Projects Agency, which was DoD focused. ARPA-H is a sister agency housed under the Health and Human Services, HHS, in partnership with the NIH. ARPA-H does not report to NIH or vice versa. They're sister agencies that are aligning their goals and objectives. ARPA-H is modeled after DARPA, where there's high risk, high reward, program manager driven efforts to impact and improve health outcomes for everyone. Recently, within the last month, 3 different announcements, openings have been announced. We have our first BAA, broad agency announcement. We're accepting abstracts for proposals for funding right now, evaluating those abstracts. So if you have innovative ideas and solutions that are can divide and identify pain points or critical aspects of healthcare system that needs to be solved, and a way to interrogate and then test where those pain points are in the system. We're looking for solutions through these abstracts through the open BAA. We also just released last week an RFI, request for information, on ideas on how to better partner with the FDA. That can really facilitate things down the road. We heard from Florian this morning about the static nature and the challenges that faces that the FDA requires of device companies, of software companies. You know, FDA is built on more historic, static chemistries for drugs or more static devices, but we're realizing more and more that devices are changing, software changes on a more regular basis, and you know, things like that could be of value to hear from the community as to how to better partner with the FDA. Likewise, we have a 64 team bracket challenge, similar to some sports bracket competitions you may be aware of. I cannot say that name, but you can go online right now, log in, and vote for your best ideas on round 1 of that 64 idea bracket. Winners, if you've played in some of these other bracket evaluation competitions, you can win money on ESPN or other things. Here you can also win money if you end up choosing the best bracket, the best teams to go forth and forward. And eventually, ARPA-H plans on funding some of those ideas that make it into the Final Four or the winning the full competition. ARPA-H is really driven by program managers, and that's part of the reason why I was brought over to a part of my time is to help recruit and evaluate and find people and evaluate the potential candidacy to become a program manager. If you're aware, you may not be, but this rugby player here was a former DARPA program manager who was the acting deputy director or acting director of ARPA-H while it was getting stood up before Dr. Renee Wegerson was called by President Biden to be the director of it. But we're actively searching for excellent program managers. These are excellent expert leaders in their field that have really brilliant ideas or are frustrated with their current situation and see that getting some funding from the NIH, from HHS through ARPA-H can really change a field, drive solutions for programs that they think are going to be innovative and revolutionary. And I know my time is running short, so I want to focus on some of the criteria that we are looking for in not only program managers, their ideas, but efforts that you would, as researchers, would submit to ARPA-H for funding, and especially highlight these last 2 points of these Heilmeyer catechism or Heilmeyer criteria. That is a historic set of questions that Dr. Heilmeyer, one of the founders of DARPA, asked all his employees to answer in generating a program. So those first 8 are historic. Question 9 and 10 were added by Dr. Wegerson in regards to the health field, because we've realized, even today and yesterday in our discussions, that health equity and accessibility cost experience is important and vital for health solutions to be implemented. We've seen that with COVID over the last few years, that also question 10, that misperceptions, how things are perceived, has a major impact on equity and how health solutions are delivered to the populace. We want to be able to reach all people and bring the health of everyone up, not just those with good health insurance, those with higher socioeconomic status, and so forth. So I will close there and take any questions, or whatever times for questions. Sorry, Ziad.
No. I mean, we have the break afterwards, so we'll bleed into that. So that's okay. We have time for one question.
Tyler, because I already know a lot of the SPARC program, but ARPA-H, a lot of people are excited and wondering, and also your technical support as well.
I can't see.
In terms of ARPA-H, obviously my understanding is that folks need to submit an abstract, but at this stage, there's not going to be kind of an interactive ability that you normally would do with NIH program managers or officers, where you could talk to them and learn more about the program. Is that correct? So it's kind of, and I guess the bandwidth of individuals at ARPA-H is still building up, right? Can you say a little bit about that?
Exactly. There's a skeleton crew, and like myself, we've been recruited to help get things stood up until they do have staff to do that. But yes, the abstract phase right now, there is no open dialogue to discuss the ideas. So it's a 3-page abstract. Submit it. We'll provide feedback as soon as possible on those ideas. At that point, the feedback on the abstract or invitation to submit a full proposal will be made. At that point, there may be opportunities for more open dialogue. It's just a shortage of staff right now, the reality is, and because it's already open, legally we can't necessarily give guidance to some people more than others at this time until we figure out the logistics of all that.
Thank you. Thank you. Thank you, Tyler. Okay, the next speaker is Dr. Chen. I also meet with her every month. And she was actually made it in a day early, and we had our site visit for Reveal, which was kind of an official really start, because we've been drowning with paperwork and felt like finally that's one step. So, Dr. Chen serves as a branch chief of the NCCIH Basic and Mechanistic Research and the Division of Extramural Research. She's overseeing fundamental science research, translational research, intervention optimization research, as well as methodology and technology development related to all complementary and integrative health approaches. Thank you.
Thank you. Thank you, Ziad, for your introduction, and I want to thank the symposium organizers for the invitation to participate in this excellent symposium on neuromodulation. My talk will focus on the NIH programs that are beyond the SPARC initiative that Tyler just discussed about earlier. So I don't know if I can fit this in 12 minutes. I have a suspicion I won't be able to join my colleague of having extended presentation. But I wanted to focus on 3 different areas for today's presentation. One is the partnership between SPARC and NCCIH, which stands for National Center for Complementary Integrative Health. If you can't remember, it's NIH plus 2 extra C's. Through the REVEAL project, and which is a combination of vagal nerve stimulation and whole person health research. And I want to go into the interception research program that NIH is developing and supporting right now with the goal to connect the brain and the SPARC, which has been sort of a big topic in yesterday's session. And then lastly, I will go through a few funding opportunities and programs that there are what we call maybe other non-invasive, minimally invasive neuromodulation programs at NCCIH, and maybe even some of them might be relevant to HEAL. So I'll start with, you've been hearing about this REVEAL project for the last few days already, and that is as Tyler mentioned earlier, it's currently SPARC initiative funding this. SPARC initiative is funding this $21 million research project too. Dr. John Osborne's team through University of Minnesota, and it's called the Research Evaluating Vagal Excitation and Anatomical Linkages. It has I have some circles there, sorry. Let me just put it all out. I think my animation was a little off. It's really a tremendous endeavor to probe implanted vagal nerve stimulation device effects on multiple internal organ systems, including the immune, cardiovascular, metabolic, and gastrointestinal systems in epilepsy and treatment-resistant major depression patients. In addition to this tremendous research scientist team assembled by John through the University of Minnesota and other institutions, it is this project is what we call a cooperative agreement. Basically means it's a collaboration between the scientists and the NIH. And so in addition to that excellent team led by John, it's also supported by a very big NIH extramural scientific and administrative team. And here's that list of NIH team members here, and I wanted to acknowledge all their contributions and efforts. And three of us, Tyler and Deborah Egan, who's been here with us for the last 3 days, and myself are part of the team. And we certainly enjoyed that site visits the day before this meeting as well. So you might wonder why NCCIH is partnering with SPARC on this, and that is because this REVEAL project fits right into NCCIH's strategic plan on promoting and supporting and promoting whole person research. And that is really, and you might wonder what that means, and we actually started that whole effort, the concept of whole person research back in 2021 with a workshop called Methodological Approaches for Whole Person Research, which we actually, through this workshop, we attempted to break down the components of this extraordinarily complex concept. And we kind of break it into 4 parts. And the REVEAL project fits what we call very nicely into that second component, which is study the impact of single component intervention on multiple interconnected systems. And that is why when SPARC called us, we said, okay, we'll work with you in administering this study. So that's that. And then but next, what I wanted to move forward, what would be beyond this project is that we actually need to, even though while we're trying to understand what the neuromodulation does to our body, and we kind of also need to understand why that might work. And that is sort of what our first component of the way we conceive whole person research is. We need to understand how the systems are interconnected. And one of such example is actually interception. And that was initially funded or supported as the NIH Blueprint Initiative on interception aimed to connect the brain and the body by filling the gaps between the brain initiative efforts that Brooke just talked about, very extensive, a very enormous program, and the very big program of SPARC. So we started with a 2-day workshop back in 2019, actually, on the NIH campus, and that workshop was called The Science of Interoception and its roles in nervous system disorders. The workshop featured 2 keynote speakers, Dr. Adam Patapodium, that was before he won the Nobel Prize, who provided an illuminating introduction on the role of sensing mechanical force in interoception, and then Dr. Hugo Critchley, who gave us a comprehensive review on the nervous system disorders impacted by dysfunctions of interoceptive processes and peripheral organs. If you haven't had a chance, I certainly encourage you to go to that website right here to check out there's a link to the workshop video recording and a lot of details about the program as well. So following that workshop, the workshop planning committee and the speakers actually worked pretty hard together and developed a special issue for Trans-NIH neuroscience, exclusively focusing on interoception. And the NIH program officials actually, because we can't publish with the speakers together, so we wrote our own paper as the opening article for that special issue on the emerging science of interoception at the NIH. Which we really decided to provide an updated definition on interoception because and in comparison to the more traditional concept, which I kind of alluded to yesterday in the Q&A session, is that we wanted to emphasize the regulation part of this concept as well. And we think it's critical because, to include the regulation when we define the concept of interoception for 2 major reasons. One is the communication, as we all know, between brain and body are bidirectional. So restricting interoception as a one-direction process will actually give a very incomplete picture and understanding of this very complex process. And then it will give us an incomplete neurorepresentation of the internal world as well. So secondly, I kind of mentioned that a little bit yesterday, is that interoceptive function is often measured by functional changes of the peripheral organs. Therefore, including this regulated component into interoception definition will be really helpful for us to develop new technologies and new ways that can help us to measure what the interoception process and outcomes or effects are. So we hope that with this updated definition, we can really help to facilitate the growth of this field. And this is actually the definition that has guided NIH development, program development on this concept of interoception. So in parallel of that, writing that paper together, we also, the program team worked pretty hard and published an RFA on interoception as the initial effort to unravel the connection between the brain and the rest of the body. And I've been told that I cannot say brain and the body because brain is part of the body. So I have to keep reminding myself that it's the rest of the body. So and you should probably be careful as well. I've been certainly questioned by the NIH Institute directors about this issue before. So by focusing on the functional and anatomical mapping between the brain and body, the RFA really trying to focus on that connection. And we sort of theorized a little bit, even though it's probably common sense, of what the process is really about. And we decided to focus on the spinal and the vagal pathways exclusively. And that doesn't mean that interoception is only those 2 pathways. It certainly includes humoral pathway as well. But because we have the limited funding, we have to make the biggest impact out of that funding. So we decided to just focus on the nerve pathways for that time being. And that's sort of the start here. And what we thought the start, if we have to start somewhere in this very complex loop, we decided to start with the ascending pathways, where it will detect and propagate the interoceptive signals in the body, and then send it through to the sensory ganglia, either through the vagal pathway or the spinal pathway in the form of dorsal root ganglion, for instance, and then further going to the brain. I think it is very important for us to realize that once it gets in the brain, it doesn't just go to the brainstem, it could go further up. into the higher order of brain structures, including insula and cingulates, but even possibly further up into the cognitive, emotional, and other sensory-motor areas. So then within then the brain is also capable, once they get those signals, capable of generating what we call regulatory signals that will then be sending down along the pathways that came up somewhat. That's what we commonly call the efferent pathways to regulate the internal organ signals, and those signals would then be further detected again. So it's this feedback loop that we could be looking at over and over again, and what we really don't know is exactly how they're wired, how the signals are detected, how they're transmitted, and how this whole loop is functioning. And that was what that initial RFA is trying to focus on, is to have a really nice mapping of those of signals. I had to cut out the grants that were funded through this because for the sake of time, but I wanted to tell you then after that, because that initiative was focused on animal models specifically due to the restricted funding we were able to generate. So we followed up with what we call a notice of special interest, and to further support interoception research. And this notice is really issued by 10 NIH institutes and centers and offices, and it supports interoception research that are more closely or directly relevant to the participating institutes' interest in specific diseases or disorders in the context of interoception, and it also supports both animal model work as well as human subject research. And this is a funding opportunity that's currently active, and it's active at least until 2024, and I have a suspicion that we will probably want to further continue it, given that we're still at a very early stage of this topic. And also this, we're very happy that somehow the Center for Scientific Review, which is a parallel agency within NIH to all the program officers institutes, had an interesting interception as well. They actually independently established a study section that's looking at the neuroscience of interception, and then they clumped it with chemical sensation. But anyway, so there is a review home there, and certainly for people who are interested in this topic, want to pursue it, this might be some a study section you want to look into more, and we certainly want to work with the review officers to make this a robust study section for the field. And I also wanted to share with everyone that within the NIH, actually, in the recent years, the program that support interception-related research most is actually the NIH's High Risk, High Reward program, actually funded through Office of Director, what we call a Common Fund project, a Common Fund initiative as well. And many of the current grants, the very high impact grants, are funded through the Director's Pioneer Award. But certainly we're starting to see applications coming through the New Innovator Award, which is targeting a little bit more in the junior phase. And then transformative R1 is another great mechanism that people could look into if you're doing very large scale transformative type of work on this topic. And certainly people should look into the Early Independent Award if you're very highly motivated graduate students at the end of graduation going to postdocs. Well, actually, you don't go to postdoc, you go directly become a faculty, and that's what it supports. So, so those are the funding opportunities. And as Brooke mentioned, the NIH Brain Initiative, in addition to that tremendous program that he described, which is highly relevant to all of you, the Brain Initiative also last year published a notice of special interest to highlight its support for research on interception circuits, certainly because it's in Brain Initiative, so it's mostly restricted to the brain circuits, but still we're just very happy that they are even interested in looking at into this and supporting this topic. And I want to highlight that this topic is so big that it is not carried by a few people. There's a large team within the NIH. We call the Trans-NIH Interception Research Working Group. This is the current roster of the members, and it consists of at least 15 NIH institutes and centers and offices, and we meet at least quarterly to discuss and coordinate interoception-related research initiatives and activities. And one example of that effort is, I want to highlight here sort of as an infomercial, that is we are planning a upcoming 2023 investigator meeting that we already decided on the date and location that will be on the NIH campus. It will be in person, open to the public for attendance. It's on November 11th. And so if you're interested, please save the date and stay tuned about registration events. It should be coming out pretty soon in a couple of weeks. And that is currently planned by a very enormous team, including both NIH program officials and a very interested investigators as well as showing in this list. Now, I wanted to switch gear a little bit now to talk about neuromodulation. Is not just invasive, it cannot only be done by invasive technologies we've heard today. There are a lot of non-invasive ways that you can modulate the nervous system. And maybe you know, maybe you don't know. One of that possible way is of interest to NCCIH is the acupuncture. And there's a few neural theories about how acupuncture might work, and there's all sorts of acupoints. We don't really know exactly what they are, what they mean, but they kind of provide a non-invasive modulation entry point potentially to us. And so in the effort of having a better understanding and maybe trying to ascertain the validity of those acupoints better, we are currently, we have a current funding opportunity which is almost ready to announce the awardee hopefully in a month or so. And that's really it's a cooperative agreement, a grant that's supporting a resource center to build an open access repository and database for anatomical and physiological correlates of acupoints. And it is an effort it will basically, this effort will aim to not only integrate very sophisticated anatomical data across the body as potential, neuromodulation points, what we call acupoints, but also it's hoping to link multiple NIH databases, such as a physiological database, such as this database built by Brain Initiative, built by SPARC Initiative, as well as the HubMap. I didn't know you were mapping how we're gonna talk about this. I'm sorry, let me go back a little bit. So that's really, and then we're trying to have an information hub about this, all the information to integrate them together, and so people can search for it in the future. So that's sort of an ongoing effort that we're hoping to launch pretty soon. And then I wanted to mention a few other NCCIH initiatives that could be related to neuromodulation. One is, neuromodulation regulated by force-based manipulations. We're actually currently funding 3 major research networks. This is in collaboration. It's actually a joint effort between NCCIH and NINDS. We're funding 3 research networks that are connecting people who are doing what we call force-based manipulation or mechanical therapy. We're trying to find the right word that would jive with everybody, but the concept is mechanical force manipulation of all over the body, and then see its impact on the nervous system. And we're trying to bring those people who are doing sort of, good examples of massage or chiropractic manipulation, or that if you can't figure out what the word force-based manipulation means, that's a good example. And we're trying to bring those practitioner type of researchers with basic scientists, very basic neuroscientists, like those studying in somatosensory neuroscience, and to bring them together. And here's the 3 networks that we're funding, and you could see each network are led by a pair of investigators. Almost one of them is that practitioner from the practitioner field, the other one is from the basic science. And they have, their job is to really facilitate collaboration, and then potentially, they have what we call the pilot funding opportunities that people can look into. They're supporting sort of pre-NIH funding activities. So I don't know if I have a lot of time going into. We actually have some Okay, so I should stop here? Okay, sorry, I think there are some that might be of interest. This is a biomarker program from HEO Initiative, and there's some workshop with that related to brain-joint connections that would be relevant to it. But other than that, I think I want to end at this slide, but I do want people to take a picture of this slide, maybe if you want to learn more about the topics that we're talking about, I'm talking about today, and get updates. I think this is a good way to stay in touch with us. Thank you. Sorry for the extended time.